KarMMa study is a pivotal evaluation of ide-cel, a BCMA-targeting CAR T-cell in relapsed refractory multiple myeloma. Ide-cel is an autologous T-cell product which is transduced with a vector that incorporates an anti-BCMA targeting molecule, 4-1BB as a co-stimulatory molecule, and CD3 zeta as a T-cell activator. So the eligibility in this study was very straightforward. Patients with 3 or more prior lines of therapy, having exposed to proteasome inhibitor, immunomodulator, and also anti-CD38 antibody, and refractory to the last cycle. Patients get leukapheresis, so you collect the cells and produce CAR T cell, which takes around 4 to 5 weeks. In between, patients can get bridging therapy if it's a more aggressive disease. 88% in this study, patients got bridging therapy. And prior to CAR infusion, patients get lymphodepletion with fludarabine and Cytoxan. And then following CAR infusion, a month later and beyond, we measured the response. 128 patients have been treated on this study, and the target dose ended up being 450 million cells. Primary endpoint was both PR and the secondary endpoint being a CR. An important point to keep in mind about this study is that the patients were very advanced disease. They were 6-year median from the diagnosis, and they had received median of 6 cycles, 6 different chemotherapeutic options before they went on the study. 30%, 35% were high-risk cytogenetics, 39% had extramedullary disease, 84 were triple refractory, 94 were refractory to anti-CD38 antibodies, so very advanced patient population. And in this patient population, the overall response rate was 73% with a CR rate of 33%. And if you look at the target dose, the response rate was 82%, CR of 39%. And a median time to response, 1 month. So it's very quick response. And if you look at the depth of response, overall, if you consider VGPR and better, 39% patients got MRD negative status. And at a target dose, 48% patients got MRD negativity. So really very good response rate in these patients. Now, if you look at who gets this response, it's almost every group, subgroup we can look at get the same response, 70-75% response rate, whether high-risk cytogenetics, extramedullary disease, triple refractory, penta-refractory, they all have very similar results. So the treatment works in almost all settings. The only place where there is a dose response is the dose of the CAR T cells. So from 150, 300 to 450, we see clear dose increase. So there is something to giving the target dose that is important. Now, duration of response was around 10.7 months. But if you look at the subgroups, patients getting 450 dose, the DOR was 11.3 months, and patients getting CR, it was 19 months. And if you look at PFS, the overall PFS was close to 9 months, but in the subgroups at 450, it was 12 months, and patients getting CR, the PFS was 20 months. Quite interesting. However, patients who did not respond, their PFS was 1.8 months, telling us that these are very advanced disease patient population. Overall survival in this study was 19.4 months. It's still early and more patients are being followed. Toxicity was, considering CAR T cells, was quite manageable. 84% patients got CRS, but only 4% patient had a grade 3 or higher CRS. And CRS median time is 1 day, so it happens very quickly. Median time of duration of CRS is 5 days. So, and patients get treated with tocilizumab. 50% patients got tocilizumab overall. Neurotoxicity is not very significant. 18% patients get it. Majority of them are grade 1 and 2. Only 3% at grade 3 neurotoxicity. They are treated either by tocilizumab or steroid. The toxicity to keep in mind is cytopenia. 90% patients got grade 3 or greater neutropenia. And it's not because of the CAR as much as because of the lymphodepletion they get. And that does put them to a risk of having infectious complications. So if I summarize this, ide-cel provides frequent, deep, and durable responses. Toxicities are manageable. And so it is a very attractive treatment in triple exposed patients with multiple myeloma. Now, the plan for future, as it becomes available in these patients with 3 and beyond cycles, is that we need to use it and explore its use in earlier setting. So in high-risk patients, that's what we are doing right now. In high-risk patients, we can evaluate what they are. We have to combine it with some other maintenance treatment. Right now, CAR T cells, one infusion and nothing else. Now we need to give maintenance so that a good response we get can be sustained longer. And then various other combinations with the drug, multiple CAR T infusions, etc., are all being explored in earlier stages of the disease, and it will be very exciting. And these results are across multiple other CAR studies. So I think CAR T cells have a great future in treating high-risk myeloma and also advanced myeloma.